You're listening to the Valley Current.
Hello.
Hey, Leonard, Jack Russo here.
Hi, how are you?
Pretty good.
Hey.
Yeah, so just tell me, what's life like for you up there? Because you're one of the people that I aspire in many ways to have a health span and lifespan matching it in a similar way. And I know you like to jokingly say, well, you have to have the right genes. My dad died at 90 a few years ago. My mom just had her 89th birthday, and she's on her way to 90. And so, you know, pretty good lifespans, pretty good health spans. They both smoked when young. They probably cut themselves down as a result of that, but by and large, they lived a fairly healthy lifestyle. But it wasn't vegan, it wasn't no smoking, and it wasn't no alcohol. So I don't know if it was truly healthy or not. But tell us a little bit about what's going on with you.
Well, as I think you know, I don't have any clinical manifestations of serious pathology, which doesn't mean that there's something going on at the molecular level that is life-threatening, but that it hasn't risen to that level yet. So I can't explain my good luck in remaining essentially pathology-free, although I do have a number of the usual age-related annoyances, but they're just at this time only annoyances. I don't know what to attribute my good luck to. I often state that it has something to do with the inheritance that I gained from my mother, who died at 106.
Wow.
My father died much earlier in his mid-70s, but he was not a highly educated man and didn't listen to what his body was telling him in his later years, and he had a antipathy to consulting with physicians. So his life probably could have been extended if he had listened to the advice of some physicians, but he didn't. In any case, the usual signs of great longevity is the absence of major health problems in earlier years. And I can attest to that observation because I've, again, luckily, have never had any major life-threatening illnesses in my younger years.
And in general, you grew up in a period where people smoked. I think the majority of the adult population, and maybe a lot of kids, my dad told me he started at the age of 14 or 15 and didn't stop until he went cold turkey at the age of about 65 or 70. So that's like 50, maybe over 50 years of smoking, including 4 packs of Lucky Strike, no-filtered cigarettes every day. And so that was the time, you may remember, when people actually thought it was sort of a healthy thing to do. At least that's the way the media at the time presented it, as though it was somehow helping your lungs. It obviously was doing something quite the opposite. So I take it you didn't smoke or have any sort of negative behaviors that might account or might otherwise suggest that you would be in trouble, right?
No, I did smoke from the age of about 17 to 30-something.
Wow.
I didn't smoke a pack a day. I smoked about 10 cigarettes a day.
I mean, that would be viewed as a lot of smoking in today's world, right?
Today, absolutely, yes. But at that time, it was sort of moderate exposure.
Right. Because people like my dad were literally chain smoking. I think a pack of cigarettes was 20 cigarettes to a pack and 10 packs to a carton, and he'd go through a carton or 2 cartons or maybe even 3 cartons a week. So we're talking about 4 packs or 80 or so cigarettes a day. If a day for him was 16 or more hours, it's a lot of cigarettes. It's like 3-4 cigarettes an hour, maybe 5 even an hour.
Yeah, I never got beyond 10 or 12 cigarettes a day.
Right. And I'm just kind of curious, when you think about your decision to stop, was it health related or cost related or something else?
Well, I had realized early on, before it became popular to stop smoking, that it was harmful. And I just quit cold turkey.
And it worked. And it worked for you for the last 60-something years since you quit, right? Or 50-something years since you quit. Because you're 92 now, right? You're going to have your 93rd birthday as your next birthday?
I'm going to have my 92nd birthday in May.
Okay, great. Congratulations. You've outlived about 99.9% of the global population.
Well, the global population maybe, but if you look at the tables from the Census Bureau, and I have those here, but it would take me a few minutes to get the actual numbers. Of a cohort of 100,000 people born in 1928, when I was born, I think there are just 2,000 or 3,000 left. But I'm not sure that number. It would take me a couple of minutes to go over to my library and get the actual data if you really wanted it. But
no, I mean, I'm just using some simple math here and say if there's a couple of thousand people in their 90s, or even call it 10,000 people in their 90s, maybe there's more, and there's something like 10 billion people on planet Earth, I think those rough numbers say you're at the pinnacle of 0.1%, or maybe even less than that actually survive with health span that's matching your lifespan, because you're taking care of yourself, you're able to walk, you're able to get up and down, wash your clothes, presumably do all the household things that people do. And I think that's even a tinier number, because so many people are holed up in nursing homes and other places other than their homes, hospitals, hospices, and the rest. I mean, no one does that math because it becomes irrelevant. But the reason I asked about smoking, is to turn to the subject at hand, which is the virus, the COVID-19 virus, and whether there are things people are doing that are effectively enhancing their likelihood of not being resilient to the virus. And I would imagine that you would say smoking would not be a preferred course for any sort of respiratory illness, right?
well, I want to go back to what you said earlier, because it's not correct. The cohort that I am a member of is a cohort born in the United States in 1928. So my cohort can't be compared with a worldwide cohort born in 1928, because my cohort is closer to living a life much more similar than it would be to compare me with a worldwide cohort. So you really can't do that. It's not fair, mainly because if you're including in the comparison all of the populations of very poor countries whose health issues are significant, and that would make my cohort or me stand out in a much greater way than it does. When you should properly compare my cohort with the U.S. Census information. And that's why I say I could easily, in 5 minutes, find out exactly how many members of my cohort are still alive, at least statistically.
Right.
And that would mean and the usual way it's done is to look at cohorts of 100,000 people. And I could easily find out the number of people, 100,000 people born in 1928, males, and how many still are alive.
Okay.
It might be a few hundred, or it might be a few thousand.
Yeah, I don't think the numbers are big, and my approximations are really back of the napkin, but the point is you've really outdone most of others in your cohort and otherwise who, rarely get as far as their 90s based on the averages. And you're headed to 100 because your mom made it to 106 in an era where there wasn't as great medical facilities as there are today, and there certainly wasn't the same degree of health literacy. And in some ways, that's kind of just background to the fact that I think you're an exceptionally brilliant person who should really be weighing in on some of what might be called the misinformation and fake news, as it were, about this virus. So I'm kind of curious, just as a way of a backgrounder, because you were the discoverer of what's now called, and it's been called for a while, the Hayflick limit. Maybe we should start there and talk about what that is in a summary way and how you think it might relate to our thinking about what's going on today.
Okay, but before I address that question, let me make one statement about my mother. Her cohort, when she died at the age of 106, she was, out of the 100,000 people born in 1906, she was maybe 2 or 3 people out of the 100,000. And when she was at that age, she had no peers. All of her friends were gone. She had no one her age or even 5 or 8 years younger than her who she knew who were still alive. And that was a very telling experience for her. All of her bridge partners were gone, et cetera, et cetera. Okay, so let's get back to the question, the last question. Do you want to know what about this Hayflick limit?
Well, how would you just, a lot of people who listen won't know or remember or have studied, and I was a biology major and a chemistry major and a biochemistry major in college, and it's a long story of how I ended up in law school. So I remember studying the Hayflick limit in college in the early 1970s, and I'm probably not going to describe it correctly. That's why I thought you should in a summary way.
You just want me to describe it?
Yeah.
I'm happy to do it.
Yep.
Well, do you want it rather detailed, or do you want it short and sweet?
Yeah, short and sweet, and then we'll get into more detail about how it bears on the way people have thought about longevity and lifespan and some of the things that affect longevity and lifespan and the like.
Okay. The discovery that I made in 1950 1959 and 1960 that later became called the Hayflick limit was an accidental discovery. It was a discovery made at a time when cell culture, or what was then more popularly called tissue culture, was reaching great popularity, and especially in the field of virology, which I won't address at this time. But the belief at the time that I did my work was that because you could take tissue from animals and plants, and animals including humans, and put the cells from that tissue into laboratory glassware, bottles, or test tubes, and then the cells would be alive and they would divide and replicate. And as they replicated and covered the floor of a flask or the wall of a test tube, when they reached the limit of what surface was available, in order to have them increase in numbers, if that's what was wanted, then there was technology for removing the cells from the surface of those vessels and putting those cells into new vessels, and then the cells would continue to divide, and you could repeat that process over and over again. And from the time the tissue culture was first discovered in 1902 until I got into the field in 1955 or 6, it was believed, in fact it was more or less of a law, that when you put cells in culture in the way I described, normal cells for the most part, the cells had the capacity to replicate or divide indefinitely. And that was what I was taught, and that's what I believed until I discovered that that was not true. But I should state parenthetically that because of that belief that when you put normal human cells into culture and they were thought to have the capacity to divide forever, that interpretation was very significant in the field of research on human aging, because if you could take cells, the tiny bit of cells from your arm, let's say, no bigger than a pinhead or half the size of a pinhead, which would constitute millions of cells, if you could take those cells and show that they could replicate indefinitely, that would mean that aging had nothing to do with what we call intracellular events, or the events that occur within cells. Because it would be obvious that if I could take cells from various parts of your body and show that they could replicate for decades, and well beyond the lifespan of a person, but certainly well beyond the replicative capacity of our normal cells in real life, then aging would have to have nothing to do with events inside of cells. It would mean that aging, and this is what was believed at the time, that aging was caused by radiation or stress or ultraviolet light. Many extracellular or events outside of cells would be the cause of aging. And that was the basis for research done at that time. When I was culturing cells, to go back to the laboratory now, and then I'll link it to the story of aging later, my efforts were directed toward understanding whether it was possible that human cancer was caused by a virus. In the late 1950s and even earlier, in respect to lower animals, There was evidence that viruses could cause cancer in mice and rats and chickens, etc. And it seemed to me that it would be logical to conclude that viruses might be the cause of cancer in humans. And in fact, years later, that was emphasized in the so-called war against cancer by Nixon. It was supported by President Nixon. And that ultimately failed, but I only mention that because that was a major understanding after I first decided to look at that question myself before the Nixon war on cancer. In any case, my experimental design was to grow cancer cells in tissue culture in bottles, and then take extracts from those cancer cells and expose normal human cells to the extracts from cancer cells to see if they could convert the normal human cells to cancer cells. The technical term for that is transformation. Well, it was very easy to get cancer tissue from operating rooms in hospitals because cancers were very common, still are, and it's easy to get that tissue, which would ordinarily be incinerated. You can get it easily from operating theaters, which I did get from the University of Pennsylvania Hospital, which was across the street from the Wistar Institute of Anatomy and Biology, where I worked at the time in the late 50s and, well, from 1958 to 1968. And the problem was to get normal human tissue to which I wanted to expose the cancer cell extracts, because humans don't give up their normal cells without serious complaint. It was also known at that time that adult humans frequently one could find normal or usual kinds of viruses in adult normal tissue, like the chickenpox virus or some other virus that would hide in the cells and not express themselves as disease at the time. So I wanted to avoid that complication of encountering normal human viruses when I was looking for cancer viruses. And so I decided to use human fetal tissue, which was known at that time to be free of such viruses. And I obtained human fetuses from legal abortions performed at the University of Pennsylvania Hospital, again across the street from where I worked, and also later on to get fetal tissue from Sweden through friends who had access to legally aborted tissue in Stockholm, Sweden. When I started the experiments, I had dozens and dozens of cancer cell populations that were growing, and I had at the same time, because it was far more difficult to get, maybe a dozen or two tissues from human fetal abortions. And when culturing the tissues of the material from human fetuses, it came a time when the cultures that were dividing happily, the cells dividing happily week after week after week, when I looked at them after 8 or 9 or 10 months, one of the cultures showed signs of not necessarily dying, but stopping their division capability. They just sat there and they didn't divide anymore.
That must have surprised you.
No, not the first time I saw it, because it was known for the previous 60 years that cells would stop dividing for hundreds of different reasons that people argued was happening, like the culture media failed, or the glassware was dirty, or the technician made some kind of mistake, and all kinds of excuses were used over the previous 60 years to explain why people's cultures died or stopped dividing. But I was in a unique position to challenge that, because although I dismissed the cessation of division in the first culture that I looked at, as the weeks went by, other cultures obtained from different fetuses were also showing loss of replicative capacity. Well, that intrigued me, because all of the cultures were being cultured by one senior technician, and the same pool of media was used in all of the several dozen cultures. So they were all handled in the same way, yet some stopped dividing and the others were flourishing. When I looked at my laboratory notebook, I then realized an important observation, that the cultures in which the cells had stopped dividing were cultures that had been replicating the longest. So cultures that I started 2 or 3 months ago from human fetal tissue were flourishing. Cultures prepared 9, 10, 11 months ago all stopped dividing. Well, that, of course, could not be attributed to the failures of a technician or the media or the glassware, because everything was common to all of the cultures. So I then decided that was such an interesting thing to try to sort out, that I put my cancer research efforts on the back burner, and in fact never did go back to that study, but wanted to find out what was going on, why these cells that were in culture longest had stopped dividing. And the major issue was, first of all, to point out that the cells that I was culturing from fetal tissue were normal cells, and every possibility of determining that these cells were normal was made, including their chromosome constitution. In other words, I had to know whether the cells were diploid, that is, having twice the number of the haploid number of chromosomes, and also whether the cells were free of any contaminating viruses or bacteria. And that, after doing a lot of other preliminary experiments to try to prove that there was no laboratory artifact causing this event, I decided to mix in culture equal numbers of cells from a male fetal tissue and from female fetal tissue. The fetal cells from the male were at about the 40th population doubling, which I knew to be very close to the end of their ability to replicate, which was about 50. And I mixed an equal number of female cells at about the 20th population doubling, so they had lots of doublings left in them. And I kept unmixed cultures of the two components carrying them on after the mixture was made. And after about 30 more doublings, I looked at the mixture and found that only the female cells were present. At that time, it was, for the first time, easy to distinguish male from female cells, by the presence of what's called the inactive X chromosome in female cells. And so that marker was important. And after the mixture divided for 30 more times, only the female cells were present, the male cells were gone, and the male cell control, in other words, the bottle that contained only male cells, had stopped dividing several weeks before I decided to look at the mixture. And the female cells were still flourishing. So clearly, if there was an artifact of culture technique, or the presence of viruses or bacteria or phases of the moon, or any other argument you would want to invoke, you would have to argue that variable could distinguish between male and female cells, which was very, very, very unlikely. So it meant to me that the cause of the cessation of division of normal human cells in culture was an intracellular phenomenon. In other words, the source of this cessation of division had to be in the cell itself and had nothing to do with anything outside of the cell. And after pondering an interpretation of what I had found and excluding every argument that I could think of, I invoked the argument, which at that time was so vague as to be a possibility, that what I was observing was senescence or aging in culture, or in vitro, as we say technically. And I decided to publish the paper. There are a lot of other ancillary stories of great interest and fascination surrounding what I just described, but I'm avoiding discussing them. But as is usual in science, when you make an observation, you write it up and you try to publish it in a journal, and if it's an important discovery, which I thought this was, since it violated the dogma of 60 years duration in the whole field of cell biology, I decided to publish it in one of the leading journals called the Journal of Experimental Medicine, where another major personality and an older scientist had been working with the same cells that I was working with, and he never saw what I saw, by the way. And so I decided to send my paper to that journal. It's called the Journal of Experimental Medicine. And after about 6 months of waiting, we received a letter that rejected my paper, arguing that what I discovered must be an artifact. It cannot be an intracellular phenomenon because the dogma was so well believed that the author of the rejection letter was a very famous scientist who 2 years later won the Nobel Prize. Well, I was just essentially a young kid at the time. I was in my late 20s, and decided to send the paper to another journal in Sweden called Experimental Cell Research. They accepted the paper immediately. It was published, and although my work was ignored and ridiculed for the following 10 years or so, my work was subsequently reproduced by many people, and so it couldn't be ignored, and it launched the field of the study of aging, giving a new insight into how aging research should be conducted. That is, you should look at what's going on inside of cells and not outside. And furthermore, the popularity of that paper became so great that it has been cited, that paper and my second paper, which was expanding the work that I did and described in the first paper, that the two papers now have been cited, which means other scientists have referenced my papers. They've been both cited more than 10,000 times. The average scientific paper of the millions that are published is cited 2 or 3 times, to give you some idea of the magnitude of the citations and their recognition. So that's essentially a very stripped down version of what happened, but it launched the field of the biology of aging.
And in many ways it does speak to about 3 or 4 or 5 different things that are maybe at play right now, which is when there is an existing view of what the science is, there's kind of a pushback on any novelty that's presented and an explanation that somehow says the existing dogma is correct. And you face that, literally what I'm hearing is, 50 odd years ago, maybe even more than that, you faced that in the way in which the initial journal, refused the publication. It's like the dogma cements the existing way of thinking, and anything new must be explained by other causes, right?
Well, as I mentioned, the dogma was so well entrenched that leading scientists in the field rejected what I had reported and couldn't understand a cause other than some mistake had been made. That lasted about 10 years, but then it became more and more acceptable, and finally became accepted universally when the molecular explanation for what I discovered came to light. In subsequent years after I made this discovery, a student of mine and I discovered that the location, that there must be, the premise that we had prior to what I'm about to say, the premise was that there must be a counting mechanism, because if I took these normal human cells and froze them in a freezer, they would remember at what doubling level they were from 1 to 50 for fetal cells. It's less for adult cells, which was another tip-off that this was telling me something about aging, because older donors, people 10, 11, 12, 30, 40, 50 years of age, etc., their cells had fewer doublings in them when you cultured them than did the cultures made from human fetal tissue. So that was another hint that I was dealing with aging. But the storage of these cells in liquid nitrogen, which is one of the coldest temperatures you can obtain easily, the cells would remember what doubling level they were. Let's say if they were fetal cells that were stored at the 30th doubling level, and in which we knew the unfrozen cells had a 50 doubling maximum. If you reconstituted the cells that were stored at the 20th or 30th doubling level, the number of doublings they had left in them after you reconstituted them was the number of doublings that it went through when stored, plus the number that would be left after reconstitution. It would equal 50. So they had a memory. They certainly didn't have a brain, but they could remember. So there must be a molecular mechanism, which we discovered to be in the nucleus of the cell, that could count the number of times the cell divided. 10 years after that discovery was made, a group of people, two ladies who were working in an entirely different field, made discoveries that came together with my discovery by accident. The accident happened when one of my, I would call the fellow a scientific grandson. He was the student of a student of mine, so I call him a scientific grandson. He was working with my cell system, and he was a Canadian fellow who was working at McMaster University in Canada, when there appeared on the scene a lady named Carol Greider, who was then working with a single-cell organism that, well, it was not at maturity single cell. But anyhow, she was working with cells that, in which they were, she and her senior advisor at the time, Elizabeth Blackburn, had discovered in this organism that the chromosomes had at the end, at the tips of the chromosomes, something that were called telomeres. And this lady, Carol Greider, came to McMaster University to see her boyfriend. And since she was a scientist, the boyfriend and others in the department at the time invited her to sit in on the weekly discussion of scientific matters. And at this meeting, she met this scientific grandson of mine named Calvin Harley. And they got to talking about each other's work, and Calvin said, you know, we could do an experiment together, each doing the part that we know best, to see whether this Hayflick limit has anything to do with telomeres. And sure enough, they got together later, did the experiment, and made the critical discovery that as the cells, the normal human cells, got closer and closer to their inability to divide, the centromeres, the telomeres, the portion of the DNA at the ends, the very tip ends of chromosomes, got shorter and shorter. And that was a critical discovery. That paper appeared in Science, one of the most common, one of the most important scientific journals, and it launched a huge interest in cell aging, the phenomenon that I discovered. And now we had a molecular explanation for my phenomenological discovery. We now know that telomeres get shorter and shorter at each division of the normal cells, and when they reach a particular short point, they trigger the cell to stop dividing. And that was very, very important. Ultimately, these two ladies who later won the Nobel Prize for the discovery that I just described, and another discovery they made, which bore on my work. That is, when I discovered that normal human cells had a finite capacity to replicate, I then said and published for the first time that only cancer cells can divide indefinitely. So normal cells are mortal, cancer cells are immortal. You could not reach that conclusion without first knowing that normal cells are mortal. because prior to that, it was thought that all cells, cancer and normal cells, were immortal. So it still remained to be determined why cancer cells are immortal. And Carol Greider and Liz Blackburn discovered an enzyme called telomerase. The enzyme is expressed by cancer cells, not expressed by normal cells. And that enzyme, telomerase, adds on the portions of lost telomeres when the cancer cell divides, so that the telomeres in cancer cells, although they do diminish to some extent, they do not diminish fully as they do in normal cells. And that explains the molecular biology of that enzyme action, which later became and still is becoming better understood, explains why cancer cells are immortal.
Tune in next time on The Valley Current.